Hello and welcome to our podcast Pager. I'm George Milner.
And I'm Stan Dale.
Tune in to hear us talk with some amazing people about the subjects changing medicine today.
From stem cells to mapping the brain and medical ethics, we hope to give you a glimpse of the bright future ahead through discussions with some of the people making it happen. Hey everyone, welcome back to another episode of the podcast. This week's episode is with Emma Rawlins. Emma is a senior group leader at the Gurdon Institute, based in Cambridge in the UK. And her lab at the Gurdon Institute is focused on questions about stem cells within the lung. How do our lungs develop from stem cells? How can these stem cell populations respond to damage and disease in adults? And can these insights be leveraged to regenerate the lung therapeutically? So during the conversation we talked about what mammalian lung development looks like, what the tools to study lung development are, how new human cell model systems are changing our understanding of development, the power and also the limitations of single-cell RNA sequencing in this field, and different strategies for regenerating the damaged lung, which you can broadly split into endogenous and cell therapy. So trying to get the cells within the lung to repair themselves and introducing new cells. And in fact, we also discussed how lung model systems are being used to better understand how the SARS-CoV-2 virus infects the airways, which felt especially topical. On another note, about the podcast more broadly, this is the first episode I'm putting out this year, and I'm really looking forward to bringing you guys more conversations with more people based in Boston this year, some of which I've already recorded, and some of which are soon to come. We're going to try and stick to putting episodes up twice a month, every other Sunday, but I think we might end up throwing a few more up if and when we have them ready. So without further ado, I hope you enjoy our episode with Emma Rawlins. Emma, thank you so much for coming on the podcast.
Hi Stan, it's great to be here.
Fantastic. Today we're going to talk kind of about your research, your lab at the Gurdon Institute, and your interests, which are all kind of revolving around the lung and how the lung develops, thinking about modelling the lung's development and how that translates maybe into new ways of treating kind of all these lung diseases. So perhaps a good place to start would be you could just tell us a bit about how you ended up doing what you're doing.
Sure, I'm happy to. I'm never quite sure what the answer to that question is. How did I get here to running a lab in Cambridge? So I grew up in Doncaster and my parents were teachers. And I came to Cambridge as an undergraduate to read chemistry, which I was useless at. But chemistry, yes, I was a failure at chemistry, definitely. But fortunately found biology and actually developmental biology. So one of my lecturers in second year, as doing natural sciences, was John Gurdon. And wonderful logical lectures, the stuff that I'd loved for chemistry, then became clear and beautiful in developmental biology. And really ended up doing part 2 genetics because I could do developmental biology.
I mean, what an amazing person to kind of stumble upon, at that kind of age. I mean, the institute you work at now is named after him. So
clearly. I mean, John Gurdon is the person who showed, actually, that, well, we could do a whole podcast on John Gurdon's work, and I'm sure you should at some point. But bring it away from John, probably, because I could go on for a long time. Let's not. So after that undergraduate degree, I sort of fell into doing a PhD because I was enjoying research, enjoying genetics so much. And having done a genetics degree as an undergraduate, I then went to study fly eye development because I'd fallen in love with fruit fly genetics. And I went up to Edinburgh and I was there for, 5 years in the end, doing that with Andrew Jarman at the University of Edinburgh. And that was a great grounding in cell biology, in genetics, and in developmental thinking. So after that, I was looking around, what should I do for a postdoc? Because I was still enjoying science, still enjoying being at the bench. And stem cells were the coming thing. And actually, I decided not to do stem cells. I decided to do vertebrate developmental biology and switch to a good developmental biology mouse lab. And I was super lucky. I wrote to a lot of famous PIs in the States. And Bridget Hogan, who had written the book on mouse developmental biology, got back to me. So I visited her, fell in love with what she was doing, which at the time was all sorts of things. A lot of work on BMP, on the skin, on the neural tube, on gonads, on early development.
So this is a lot of sort of body patterning
Yeah, exactly.
and axis.
So fundamental developmental biology questions that could be answered with mouse genetics. That when I joined her lab sort of a year and a half later, she said she'd just moved to Duke to be head of cell biology. And she said, I'm going to focus, Emma. You can do anything you like, but it's got to be in the lung because I'm going to focus my lab. And she was totally in love with stem cells. And what I ended up doing was asking which cells in the mouse adult lung, as well as the developing lung, were truly the stem cells, using classic developmental biology techniques. And that was really the first time those techniques had been applied to the lung and to lung disease. So it was a really fun time. So we're setting up lung disease models, lung injury models, and building all these different transgenic mice. So I was there for 5 years, but we had a number of papers, a number of really exciting papers, and we made a lot of very fundamental discoveries.
Fantastic, yeah. So at this point, you're thinking about the lung, you're thinking about stem cells and stem cell populations within the developing lung and maybe the adult lung in these mouse models. How does your postdoc lead to you ending up in Cambridge again?
I married to a Brit, so my husband and I were British, and we went to North Carolina thinking it would stay a couple of years. Ended up staying 5. At the end of 5 years, we actually had a 3-month-old, and it was time to come home and be closer to family. So I just started applying for jobs in the UK and was very lucky when the Gurdon Institute in Cambridge offered to host my fellowship. So essentially a career development fellowship, which I wrote and got, and was hosted at the Gurdon Institute.
Yeah, I mean, it goes to show you, like, a successful career requires kind of breakthroughs at several points, I suppose. Great. Okay. Well, why don't I think now would be a good time to think about, as you know, the history of this area of research has developed. How has our understanding of the lung changed, and what systems have been useful for understanding what's going on, and sort of what is the historical perspective on human lung development, and also what are the stages of the human lung development? Yeah. So
let's start with that. So actually, the historical perspective on human lung development is stages. So one of the first papers that anyone coming to the field reads is from a Dutch scientist, and I don't remember when it was authored, I want to say in the 60s, describing histologically, so under the microscope, what different stages of the embryonic human lung look like. And they were named as pseudoglandular, canalicular, saccular, and alveolar. I confess I hate the names, because they don't mean anything to anyone outside the field, but it's a very beautiful piece of work that's actually stood the test of time, just describing, based on the morphology, what the lung looks like. But essentially what that means is you start by your lung grows by branching. We call it branching morphogenesis. And it branches to make the airways, the tubes that the air goes in and out of, and then it branches to make the alveolar sacs, which is the gas exchange surface. And until very recently, we essentially had no clue, apart from the morphology, how that happens in a human. All the work was based on mouse models.
So the idea being that you would basically look at what was going on in a mouse and try and extrapolate that to the human situation as best you could.
Essentially, actually, mice are a fantastic model. I think we all like to diss them now because we're excited that we can grow human cells, which I'm sure we'll come to. But mice are a great model because vertebrate development is essentially conserved. The signaling pathways that you use are conserved, most of the transcription factors are conserved, and almost all the cell types are conserved. Not precisely, but enough to get a very basic understanding, or even more than a basic understanding, a very detailed understanding.
So it will continue to be sort of a good adjunct or comparative model for looking at what might be found in these newer in vitro kind of human models, which people are so excited about.
Exactly. And that, I think, will continue for quite some time.
So how did the field go from thinking about mice to now, everyone's doing things in human cells, whether they be organoid models or on alginate beads? There seems to be a whole sort of array of different things people are doing.
So that change happened, I think, concomitantly in a lot of different fields. So not just lung development, but if you work on the kidney or if you work on the brain or any other structure. We've known for years that treatments developed to mouse models of disease often, but usually don't cross over perfectly to human. Mostly they fail. So we've known for years things were basically conserved in mice. But if you're talking about designing therapies and really detailed molecular mechanisms, you needed the human mechanism. But throughout most of my working life, the only human cells we had access to in a dish were cell lines. So usually cancer cells, very, very genetically messed up, and really not resembling whatever organ they came from. So the lung cell lines really don't look much like lung cells. And that changed when I was a postdoc with really two things. So one thing was called Yamanaka, developing iPS cells, and the realization that we could follow developmental mechanisms in order to differentiate iPS cells to essentially whatever cell type we like. But the second key thing, I think really the key thing for me, was the work of Hans Kleber's lab in Utrecht, where they showed you could take adult mouse stem cells and grow them in what they called organoids in 3D culture in a dish. And at the time, this was just a stem cell assay. This was just proving the cells they'd found in the intestine really were stem cells. And now it's become a whole way of growing human tissues in a dish in order to do much more directed either developmental biology or disease modelling research.
That's very interesting, and in a sense, perhaps I'm oversimplifying this, but the first discovery we're talking about is thinking about how can we manipulate what's going on within the cell to more closely resemble what happens in development now that we have these iPS cells. How can we introduce factors to generate cell lines we're interested in? And the second one is really thinking about, well, what are the spatial components of tissues and how might that be important in directing the growth of different cells? So there's sort of two edges which allow you to kind of gain a proper insight into
exactly right.
What's going on.
And of course, the other thing that people have now done is combine the iPS work with organoids. So you differentiate your iPS cells until they've got the cell type you want, and then you grow them as an organoid 3D culture. So you've really got to have both. And I think a really coming area, something that we're trying to get into by collaboration, is really bioengineering in order to improve all of these spatial cues and spatial and mechanical cues we can give to our cells growing in the dish.
I think this is a really good point. I mean, is it as exciting as organoids are? I think maybe this is a bit of a harsh analogy, but you could almost compare them to like some sort of abstract Picasso representation of a photograph, right? Like they might resemble an organ on a macro scale, but a lot of the fine details of what's actually going on in human development might not be there.
I think that's exactly true. But if you're comparing the organoids to a Picasso, then I think you have to compare what we had previously to some sort of cave painting on a cave wall. So we've come a long way, and we're going to make it much better.
It's come a long way. And I suppose this is where some of the other model systems... Oh, I don't want to jump ahead, but this is where some of the other model systems might be useful for kind of verifying what's seen in these
this is why
models.
We still need the in vivo... That's why we still need the in vivo animal models. And the question is really how to make best use of these now. Now we have these human cells. And this is something I think we're all still struggling with as a field. So the mice are wonderful because we can do such great genetics. They have a rapid generation time. We can grow them quickly, really in purpose-built mouse houses in the lab, or usually below the lab underground. But are they the best model? Now we've got this wonderful genetics. Should we be thinking about sheep models, for example? And if you're interested in lung development, well, sheep have very similar lungs. And actually, they've been used for years as a model for premature birth. So you can deliver lambs early and put them on a human-sized incubator and study the effects of prematurity and the treatments given to premature infants using a much better animal model than you can with a mouse. So should we be thinking about this sort of thing for more complex lung diseases as well? And I think we're all...
Yeah, I suppose also it depends on whether you're interested in looking at normal development or different kinds of adult diseases. Let me sort of pose a hypothetical. Do you think in 10, 15 years' time, there will be quite well-established, not protocols, but norms in terms of, well, we found something and we tested it in this model system, and then we advanced it through these sort of two or three other steps to show the finding, rather than right now it seems a bit more like the Wild West in terms of what's validating.
Yeah, it definitely is the Wild West. I think it's, I hope in 10 years it's more... more defined, more concrete. We have more of a plan as to what we should do. But I think we're still at the stage where we have to try lots of things to work out what the most appropriate one is. And I don't think there's going to be one right answer either. Just like there's no one right in vitro cell model at the moment, there's going to be no one right in vivo model either. And it's going to depend very much on exactly what you want to model, whether it be cancer or an aging-related disease. So there's, a lot of work to be done.
Well, that's a shame. Here I was thinking that it would be simple in 5-10 years' time.
No, I think it's going to be bespoke. And now we can have such great genetics in many, many species. Then really, you can, in theory, imagine doing bespoke models for all sorts of different things.
Let's go back to the in vitro differentiations for a moment, because I think it's worth pausing on these and thinking about how remarkable they really are. We can take iPSCs, which have been reprogrammed from some somatic cell in the body, and quite literally replicate, to some extent, what's going on in a growing organism to create these cells. And how people come up with this stuff, I know people did some sort of screening process to see what transcription factors were activated by what, but it really is quite remarkable to take a cell through, for example, with the lung, endoderm and then different progenitors to produce different lung cells. I think that's just, it's really amazing. I mean, where's my question? I suppose one of the questions I like to ask off the back of that thought is generating some parts of the airway easier than others? I mean, perhaps the components which are at the bottom involved in gas exchange might require more kind of spatial cues, because you think about the structure of that, it's very closely tied to the blood supply and
that's
and that
a really great comment. So at the moment, it's definitely easier to make airway rather than to make the gas exchange, the alveolar surface. But it's quite interesting, and some of our own data sort of plays into this. So you start with this immature cell that we think resembles a very, very early cell in a human embryo. As you say, differentiate to endoderm, then to anterior endoderm, then to ventral anterior endoderm, then eventually into foregut of the lung cell you want. But then you have a lung progenitor, which can make either airway or alveoli. And depending on what cue you give it at that stage is how you tip the balance. And it is easier to get airway at the moment. I think largely because we've been growing adult airway cells in a dish for many years. More than 20 years now. So we have some experience there. So we knew what we were aiming for and where to go. Until quite recently, nobody could reliably grow an adult alveolar cell. And I think the alveolar development is still the weakest part. So as you said, you've really got to have a very close relationship between your epithelium and your vasculature. That's the whole point of this tissue, to facilitate the gas exchange. But it's incredibly hard to get the endothelial cells, the vascular cells, to grow in the cultures. So we can make now type 2 and type 1 embryonic lung epithelial alveolar cells, but we can't bring into close composition, spatial proximity in vitro with the endothelial cells next. That's really the next challenge.
And I suppose this comes back to the word bioengineering. This is where growing something on maybe some kind of graft where you can put blood vessels in more easily.
This is certainly one of the techniques.
Or would come in handy.
So you may have heard sort of on a chip, lung on a chip culture, heart on a chip, whole body on a chip, some people are trying to do. So can you put for example, your endothelial cells on one side of the membrane and your epithelial cells on the other and get them to talk to each other within a microfluidic device? But unfortunately, the endothelial cells are incredibly difficult to grow. We've learned that to our own cost. We've spent quite some time messing with it and failing so far. It's coming. It will come. It can be quite unsettling. The endothelial cells, I think, are remarkably plastic, and they tend to de-differentiate to fibroblasts, even with these more sophisticated cultures at the moment. So getting this to work is going to be a real challenge.
I wonder if you could set up some sort of signaling loci, like if you knew a certain signal was important for keeping them as the endothelium, maybe you could have some genetically engineered cells to just produce factors that do that on the
based
chip.
on the work of... No,
I was worried you were going to say that's
it's a
completely
great idea.
bonkers. But I'm glad.
We tried very hard to do something similar to that, and we're unsuccessful. But I think we've not quite got the right factors yet. So something that we're still doing a lot of in our lab is actually a lot of description. Sometimes developmental biologists are accused of being stamp collectors and just describing things. But actually that description should give us description of the human lung, description of the mouse lung in development, should give us an idea of what those signaling cues should be, and then we can test those.
I suppose building stuff without knowing what's already going on is kind of doing so blindly, so people shouldn't knock just kind of describing stuff.
It is, and describing stuff has definitely come back into fashion. There's a lot of describing.
Okay. Yeah. Well, speaking of which, why don't we dive into some of the techniques? Single cell RNA sequencing. That's a hot thing. We were talking briefly off the air about the kind of human lung cell atlas that came out maybe a few years ago. An attempt to kind of describe all the different cell types that might be there in the lung. Single cell RNA sequencing, something that's very useful.
I think the ultimate in stamp collecting, and developmental biologists love it. So, but not just developmental biologists. So I'm sure you know the Human Cell Atlas project is a project that's set up to define every cell type in the human body. Ideally in normal homeostasis, in development, and in disease. So how do things change in disease? Where do they come from in development? What signaling molecules are talking to what in the entire body? And in theory, this is going to allow us to then model all of this much better in vitro and do our functional work much, much better. But the Lung Cell Atlas is certainly not done. It's come out and it's ongoing. I think it's one of the foremost ones in terms of organ systems that are being atlased. But there's an awful lot of people all over the world continuing to work on this. So quite exciting. I think it's 2 years ago now, we found, based on the mouse lung, new cell types that we didn't know about. So brush cells had sort of been described, but not very clearly. And ionocytes were named, I think, 2 or 3 years ago, based on single cell sequencing of the mouse lung, a cell population nobody had seen before because it's so rare. And it turns out to be the cell that expresses most of the gene called CFTR, cystic fibrosis transmembrane receptor. So then now the key question is, are these cells key to the disease CF? Or are the other cells that express very low levels of CFTR actually just as important? And that's of course something.
I wonder if you could do some sort of knockout study in a cell-specific manner to answer that question.
Exactly, and there's people doing that. So we're waiting on the answers.
But that's very exciting. And I suppose from a regenerative point of view, there might be some rare cell types which would be lost in kind of bulk RNA sequencing, which could be important for regeneration.
So we're all very much looking for that. But actually, from a regenerative point of view, what we can also get is cell states. So single cell sequencing doesn't just give you cell types, but your cell state after an infection or after an injury. And how does it respond to that in order to repair the lung is actually probably more important, I think, at this point than the rare cell types.
In a sense, you can look at differential cell responses when a given insult occurs.
So how much is common, how much is different. And then what's becoming very exciting is these transient things we think we see in normal regeneration possibly persist in cancer. So again, very, very early stage, but the more sequencing we do and the more description we do, the better of a handle we're going to have on this.
Explain that in a bit more detail, if you wouldn't mind.
No, of course. So a good colleague of mine, actually from the Stem Cell Institute in Cambridge, Ji-Hun Lee, just has a very, very nice mouse paper that came out, I think, early this year in Cell Stem Cell. So what they showed was that in response to an injury, the type 2 cells in the lung, so the alveolar epithelial cells, essentially de-differentiate slightly. So what they've showed is that after an injury, the type 2 alveolar epithelial cells, which at steady state make important surfactant proteins, but just sort of sit around and do nothing. After the injury, they start to de-differentiate slightly, take on characteristics of a type 1 cell, and they've described sort of 3 intermediate states that get you to this type 1 cell. But interestingly, what it's beginning to look like is that in a lung cancer, or sometimes a fibrotic lung, you can find cells that are trapped in one of these intermediate states. So they've sort of entered a regenerative state repair process, but they can't finish it and they can't differentiate. So possibly they're just keeping proliferating, keeping producing lots of cytokines to call in immune cells, and they're trapped in this state. This is how we're sort of currently thinking about various different lung diseases.
So essentially, just to give the listener some perspective, these type 1 cells are the cells which are actually providing most of the surface area for gas exchange, and these type 2 cells, you knock them slightly saying they're not doing much, but they're producing this they're producing the surfactant, which is important in reducing that surface tension so that you can breathe a bit more easily, and that's what premature babies sort of struggle with.
Exactly. So why I say they're not doing very much, just to backtrack on that slightly, is that the lung doesn't really divide at steady state. Lung cells sit around, they do their important adult mature jobs, but they don't really divide. And if you compare that to some other tissues like the skin or the intestine, you've got dedicated stem cells in there that are constantly dividing to constantly repair cells, whereas the lung doesn't seem to bother until it suffers an injury.
And I think it's fascinating. So what you're saying is some of these cells will get part of the way through this process of differentiating into the type 1 cell, get confused and just end up not really doing anything useful and producing all these pro-inflammatory factors which are going to pull in the immune system and just lead to fibrosis and other problems.
No, that's brilliant. It's a really great description. But what we don't know yet is how that happens. So we've got the description. We've got this recent description of a normal regeneration process. And we have a description of these aberrant cell states at an end stage of a disease. But what we don't know is how you got from the normal regeneration to this very chronic disease.
So we spoke a bit about RNA sequencing. I mean, there's a whole host of techniques we could talk about. We probably don't have time to dive into all of them. Are there any in particular you think are interesting, which provide new insights into in vitro stuff?
So the RNA sequencing has been brilliant, but what it does is it explodes the lung. So we dissociate the lung or any tissue to single cells. We then sequence what their current state is based on what genes they have turned on. But then you've got to work out how to put them all back together. So where does this cell population that I ever found actually sit in the lung? And it's something we're spending a lot of time doing in the lab at the moment. We've been doing a lot of our own single-cell RNA sequencing experiments with collaborators at the Sanger Institute. But what would be really great is not to do the exploding step and to get the spatial information at the same time as we get the detailed transcriptome information. And that's the technique that's really coming. So in situ sequencing, so looking at many, many genes at a single cell resolution, but within the context of a tissue section. So you can see if you're looking at an airway cell, a vascular cell, an alveolar cell, and actually where they are. So this is something I'm very much looking forward to. It's a very, very new technique, but it will be amazing.
How do you do that?
I'm not sure, kind of sure I know, to be honest. No, essentially you have a pre-designed probe set that's hybridized to a glass slide and hybridized very, very tightly. So you cut a fresh tissue section, stick it on that slide, and you can actually see within a single cell which of those probes light up and are active. So it's a next generation sequencing reaction, but now localized into a tiny fraction of a glass slide. How that is that is interpreted by bioinformaticians is well beyond my understanding. The data that are coming out are very beautiful. The person who's been really pioneering this is one called Long Kai at Stanford, doing some very, very beautiful work.
Well, I'm going to follow your advice, which you gave me off the air, and get started on the
bioinformatics
ASAP. So I think what you're describing is similar to what I, again, off the air, described to you as like slide seek. But the salient fact is that what we're saying is the RNA information is great, but what's important as well is where these cells are being what they're being.
Which cell is talking to which other
to the
cell.
nearby cells.
And how does that go wrong in disease? And how can we model it in a dish?
Fantastic. Well, let's get on to something which I think is very topical, and I'm really looking forward to asking you a bit about from a complete sort of place of ignorance. I don't know anything about this, but currently we're in the middle of a pandemic with a virus which infects us, through a respiratory mechanism and is famous for affecting the lungs most significantly. How are some of these lung model systems we have? Are any of them being used to change how we understand COVID?
Oh, completely. You can imagine that a lot of people who are growing these cells, and as soon as COVID hit, that's one of the first things they've been starting to do is to ask, well, can I help? Can I make a difference to the pandemic? And I guess in practice we probably can't, as lab biologists. It's the people developing the vaccines that make the difference. But of course, what we think we're doing as cell biologists, stem cell biologists, is now providing a system that can be used to study any virus. So it's a very exciting time. So if we think about COVID and actually any sort of respiratory virus that's passed on by a cough or a sneeze, you tend to get an infection in your upper airway tract. Then that can spread down to your lung into the alveoli. And it's when you get a very bad alveolar infection that essentially you destroy the epithelium, the virus can enter the vasculature and go systemically. That's when you become very, very sick. So people have been using really two types of models. Just a very classic, very old model of growing bronchial cells, or what we call an air-liquid interface culture. So the outside of the cell is the apical surface is exposed to the air, and the basal side is fed by medium from below. And you can just drop the virus onto there and look at which cells are infected, what gene expression changes as the cells are infected, how does the infected virus change? And that's already very exciting. So it seems that it's ciliated cells in the airway that are quite possibly the initial site of infection. But then what's been historically very, very difficult is to grow adult alveolar cells, adult human alveolar cells, for asking questions about infection or other sites of disease modelling. And then 3 independent labs during the pandemic have now, they were already working on it obviously, but have now perfected as well as they can at the moment their alveolar growth systems and infected the cultures with the virus. And all of those are organoid models. So growing lung cells within a sort of lump of extracellular matrix called matrigel. And then you have to sort of get the cells out in order to infect them with the virus. And again, we can ask questions about, I haven't done it, but different labs are asking questions about how the cells are infected, what sort of cytokines they turn on to speak to the immune system, what are the transcriptional changes they make in terms of their response to that infection.
Those are really exciting experiments to be doing, and the fact that it might be generalizable to kind of future pandemics might be a bit dramatic, but this is something you could use to look at all sorts of different viruses, is really amazing. I mean, you think about the lung in one sense, it's like if the human body is kind of a fortress against pathogens, it's the open door. I mean, all of that blood, all that surface area sitting at the bottom of the airway is, in one sense, the easiest way for us to be attacked from the outside. And so understanding how that might happen with all sorts of different pathogens is, really exciting. I wonder, have any of the kind of findings about how COVID is affecting these epithelial cells or cells in the alveoli, have any of them kind of been translated into anything in terms of rethinking clinical treatments?
And I don't think they were in the short term. But what I think is really the potential of these systems is that now we have a real cell type that can be infected. So previously, we've been infecting various cell lines that we're referring to as cave painting earlier with virus. And often we're taking the cell line, expressing this ACE2, the virus receptor, and then infecting cells. And the good thing about that is the cell lines grow wonderfully, and you can do lots of things like a CRISPR screen to understand how the virus gets in and what sort of pathways we can target to stop that happening. But what we're faced with then is the problem of how will any of that translate to the lung. So now we have the potential of taking those screens that have been done in a simple system, finding the targets, and asking which of those work in these adult alveolar cells that we can now grow in the dish. Unfortunately, the adult lung cells still grow very, very slowly. The fact they grow at all is amazing. So doing the whole genome CRISPR screen in the adult cells would be difficult. But if we could take the cell line work and just retest the hits in the adult cells, and this really has the potential for finding drugs possibly that can be given at an early stage of infection to stop it getting too bad. So to slow down the subsequent infection of the alveoli, for example.
So from a screening perspective, in that sense, it lends you something which is much more high throughput, and then also a further model which is much more accurate necessarily than anything we've had before to take a look at the things you find earlier.
I think it should be
really
wonderful doing lots of things.
Yeah.
But I'm just going to raise one point that you describe the lung as sort of this open door for infection, but it's a really well defended door.
I'm sorry to have insulted
really well
by
defended. So it has a lot of resident immune cells just sat waiting to attack pathogens that come in. And we talked about this type 2 cells earlier secreting surfactant to lower the surface tension, which they do. But they also secrete surfactant proteins that act to mop up bacteria. So even the type 2 cells have a host immune response.
Well, you make a very good point. I'm going to bash the lung from another angle now and say, I wonder if the high degree of immune cells resident to the organ can sometimes work against the organ in terms of regeneration. Can you get quite chronically inflamed states within the lung and overactive immune cells? And I think this is not to commend myself too much, but I think this is a decent segue into thinking about regenerating the lung. I don't know where to begin.
Well, let's begin with the problem. So there are chronic lung diseases where essentially the lung becomes destroyed. So you lose your gas exchange surface, either because it's just gradually becoming simplified and the cells are not being replaced, or because it becomes what we call fibrotic and the normal gas exchange surface gets clogged up by lots of just mesenchyme cells sitting there that is probably an aberrant regenerative response. But I think a key thing to point out is that it's amazing how often this doesn't happen. And the current thinking is that for these really chronic diseases to develop, we're talking about decades of productive injury repair that eventually becomes unworkable. So it's amazing, I think, for how long the process can work before it becomes exhausted.
And I suppose when we think about regenerating the lung, there are fundamentally two approaches you could take. You could think about giving some sort of cells from outside the body, some exogenous allogeneic cell graft or autologous cell graft, or the alternative would be stimulating the lung's own ability to regenerate itself. So maybe we could just dive into what the pros and cons of each of those approaches might be. And I sort of have a suspicion that you're going to talk about how it might be easier to get the lung to do it itself, which I think makes sense when you think about the lung's architecture. Sort of actually quite a hard thing to transplant.
Yeah. As a developmental biologist, then yes, how we get the lung to do it itself, I think, is the most exciting question. So as we're describing better and better, and we're modelling better and better human lung stem cell states in vitro and in vivo, then can we find the right cue that we could then give topically, for example, by an inhaler, to get lung stem cells to proliferate? So that's the other beauty of the lung. We work in something where you can deliver drugs to it in really quite a straightforward way. So my favourite hypothesis is that what we need to do is get the lung stem cells to reactivate in a controlled fashion. So you want them to regrow often an alveolar state or regrow a specific alveolar unit, which is why I'm so excited about developmental biology. Because the thing about normal development is that the lung builds itself, it makes its own matrix, it controls its own cell-cell interactions, and then it stops. And you don't end up with a cancer. So could we stimulate one of these normal self-limiting processes? This is my dream. And I don't know, maybe by the end of my career, I will know whether or not that's possible.
I mean, it certainly seems more attractive to, let's get the lung to do again what it did before than try and do it all ourselves, because the lung's probably doing it more intelligently, quote unquote, than we could.
But the other approach is this cell therapy approach. So could we take exogenous stem cells, and I guess the best source of exogenous stem cells is probably going to be iPS cells that are differentiated to lung cell types. And could we somehow get them in? And actually, you can do that in a mouse. But to do it in a mouse, you have to injure the lung to make some space so you can put the extra cells in. So I find that quite a scary thought. So if you want to injure somebody with lung disease to give them new cells, maybe the injury would be too much. But the other approach that's been tried is to deliver the cells through the circulation system. So if you think about your circulation, the entire output of your right heart has to go to your lung. So if you get cells into the circulation, they will pass through the lung in the circulation. So if you can get them to leave the circulation and enter the tissue, so far that's really inefficient. But if you could, then that might be a viable way of doing it.
And presumably cells which end up seeding other sites will sort of die because they're not adapted to it.
That's the theory, so long as you haven't accidentally made a cancer cell in your dish, I guess, which would always be the concern.
Yeah.
But I think cell therapy is probably going to be a lot more tractable for other tissues.
Well, I mean, for example, I'm working on stuff like in the pancreas with beta cells, and I'm just having this conversation is making me realise that the problem my lab is facing is with respect to a cell therapy. It's really going to be much easier to do something with like an endocrine cell you can put anywhere than with the lung, which has this very specific architecture and cells have to be in the right place. So it's a really different problem. It sounds to me like it might be a bit easier perhaps to just get the lung to regenerate itself.
But then maybe in the end stage lung disease, maybe the cells are too exhausted. So we don't know yet. I think it's definitely worthwhile from a research point of view of having both strategies going ahead, and we'll see which is right. And I think the other key thing is that with some well-designed experiments, you learn from each one. Not only is your therapeutic approach viable, but you learn some fundamental biology that will help design the next attempt.
Fantastic. Well, I think that's all the kind of the things I had in mind to talk about. It's been a real pleasure speaking. Thank you for taking the time.
You're welcome. It's been a lot of fun talking to you.
Thank you for listening to the Pager podcast. If you've enjoyed it, then head to Spotify or iTunes to hear more.
As ever, we'd love to hear what you think, and you can reach us via our Facebook page or by email. See you next time.